All right, we'll just get started. Let's see if anybody else comes in later. But welcome to week 2 of Bio100 Lab. Hope you guys had a good first week of school. This is what we're going to be going over today. First thing, we're going to go through the ecosphere stuff. Show you some videos on how to set it up and collect samples. I'll run through the project itself for a little. If you guys have any questions about that, we'll go over that as well. Then we're going to talk about iNaturalist for a little bit. See how you guys did last week. Tell you about what we're going to be doing for the future in that. I'm going to have a real quick lecture on biomolecules and pH. We got sourdough starter project at the end. I'm going to introduce that. And you'll have some stuff you kind of need to get ready for that as well. First assignment is due tonight by midnight, so if you haven't taken your 5 observations yet for iNat, please get on that. And I'll talk about that more when we get to the iNaturalist stuff. And then we'll deal with sourdough when we get there. So let's start with the ecosphere. All right, so I briefly talked about the ecosphere last week, and I just made mine this weekend. But it'll end up looking something like this. I know Ayub just showed me his. He's already made his as well. So it's a little self-contained ecosystem, completely sealed, and will be the entire semester. So you've got to make sure you follow the procedure right, so you get the right gases in there. I feel this blowing in the water for a little bit. And it should stay alive if you keep it in a well-lit place. If things start dying, we are going to make a second one in about a month. So before we get there, we're going to be formulating hypotheses on things you could change in it to maybe make it last longer, or if it's doing well, maybe just some things you want to change about it and see differently. I sent out a thing with the supplies, so everybody should have gathered those by now. If not, that's okay, but it is going to be due, your setup is going to be due next week. You're going to have to submit a selfie of you with your ecosphere, as well as your initial observations and measurements, and I'll go over those in a sec. But let's start with a couple videos. Our coordinator took some shots of him collecting materials as well as putting them in a jar. So we're going to watch both of those and then go over the procedure document. Oh, wait.
Hey, Bio100 General Bio people, if you're in lab and you need to do an ecosphere, I'm going to be showing you some stuff. We've got a little creek right down here right off campus, and I'm going to show you some tips on how you can collect what you need. So stay tuned. Hey guys, first a quick tip. You got a milk jug, then you possibly have a water scoop and a funnel built in one. So let me show you something. This will be easier if we take the lid off for a little bit. Squeeze this. Do a quick cut with some scissors, and I'm just going to cut out a fairly big hole that I can use to scoop water and maybe, I don't know, some sand or gravel. Boom, hole. Okay, now I probably should have cleaned the milk out of that first. What I'm going to end up doing is going down there, scooping out like this, and then I'll be able to turn it like this and pour it like a funnel. Check it out. Hey guys, let me show you something really quick. You see all these cool aquatic plants down here? Well, some of those you might want to know what they are, and one of the things you could do is you could use your phone, shoot a pic, and then put it on your iNaturalist. Then you hit suggestions, and it may make guesses, and other people will pop up later and say, yeah, that's an aquatic plant with the name such and such, right? And so even though it's unknown, we're going to get some for the ecosphere. It's not part of the project to do your iNat, but have your phone with you and take some shots. It'll help you. Okay guys, I've got my handy milk jug cut out, and I'm going to get right down in here and just use it to scoop some water. And we know this is good creek water. And then, with what I don't spill, I'll be using that other end as a funnel. Okay? Hang on. Here's what I'm looking at the bottom of this creek near campus. I'm going to try and get some of that algae mat that's floating there. I may try to get some of this aquatic plant that's right below me.
Yeah, some of that. And then I'll stick my hand in and just get some slimy, nasty stuff from the bottom, some gravel. If you don't want to do that, you can use a garden trowel or something.
All right, so that was the first one. And guys, one of him setting it up.
Hi guys, we're back. I've got all the stuff that we collected, all the plants and substrate and water from the stream ready to go, and we're going to build our ecosphere. I've got this nice 1 gallon jar. We're not going to fill it all the way. We're going to leave a nice gap at the top for gas exchange, so CO2 and oxygen could go between this little air pocket and the water. The jar I got had a hole in the lid, so you probably can't see it, but I've taped over both sides of it. So that should do enough to restrict the exchange of gas through the lid. So this will be, we'll consider it a completely sealed system. So I'm going to take out my substrate, and because the stream I was working on was flowing a lot faster than I thought it would be, a lot of our substrate is pretty rocky, and so you got to be really careful. Don't want to break the glass. And there wasn't as much mud as I kind of hoped to get. It looks kind of muddy and sandy going down the side. I'm just going to go ahead and rinse a little bit with stream water. Okay, and that's going to make it look kind of muddy and nasty again, but that's okay. Rinse down the sides a little bit, get a little bit of water in here. But I'll tell you, in the time it took to get back here and get set up, the water settled quite a bit. So that's probably because the water was flowing so much. All the really, really small particles that you might get in a more muddy, slow-moving stream had been carried away. So what I'm going to do now is just take out some of these aquatic plants using some metal tongs from my kitchen. I'm definitely going to clean the heck out of these tongs afterwards, because you couldn't smell it, but that whole stream environment was kind of nasty where we were. Okay, big old section of plants. And you probably noticed that I covered my table. I didn't really want to be dealing. Oh, there's a leaf. I'm going to put that in. It may break down. It may get colonized by something that wants to eat on it. We don't know. Give this a swirl. I think I got some more gravel in there. I do. So I'm going to add all the liquid from my plant samples. Looks like I got some more substrate. Whew! Okay, and you know at the beginning when I tell you to analyze the smell, one of the options is rotten eggs. Check. Right away I could see on the top, I don't know if you guys could see it, some little green bits floating. That's duckweed. So we have an aquatic plant already. It actually floats to the surface. Got more little substrate here. I'm going to pile that in gently. And a couple of rough little rocks. I like the rough ones better. No comment. They give more surface for stuff to attach to or more. You know how if you're painting you might roughen up a surface so that the next layer of paint would adhere to the first, and we got a little bit of that going on. So I'm just going to top this up to about there. Almost done. And then I'm going to grab a clean napkin, wipe this off, and this is the weirdest thing, but this should help us a little bit. What the heck was I doing? Well, you know, we're animals, and we engage in cellular respiration, which means that when we do our breaths, or our whole body respiration, we are exhaling carbon dioxide. So I just put a bunch of carbon dioxide in there, and that should be beneficial for the plants. A lot of people think that that displaces oxygen or something. It doesn't. So whatever oxygen was in here, whatever oxygenation was increased by the pouring action, that's still there. I just added some CO2 to it. All right, well, It's pretty cloudy. I think you guys could see, and then we call that turbidity. So one of the things you have to assess is its turbidity, but you're going to come back later and check it again. So when you first set this up, allow for some time to come back, maybe 4 hours later, certainly the next day. So don't wait a week to check on this for your second time. One of the things I'm going to do too is I'm going to use my phone app called Light Meter and get an exposure of how much light is here. But this isn't actually where I'm going to leave it, so I'm going to do that right where it's going to sit. All right, guys. Get in contact with your instructor or with me, the coordinator, Professor Joe, and let me know if you have any questions.
All right. So those are a couple of good videos on getting your ecosphere set up, and the links are on our Canvas page. Somebody asked, does our jar need to be that size roughly? That's a good size. It doesn't need to be that big if you can't find a gallon jar. Abe, do you still have yours sitting next to you? Yeah, can you hold that up? Yours is a good size. Like, that's totally acceptable. It doesn't need to be huge. Way to be ahead of the game there and having that done. Let's go to this. Walk through a couple more things. All right, so I put this up on Canvas as well. It's the actual written instructions for your ecosphere. You need all these things. As far as the source of water goes, if you're near San Diego State right now, there's a little creek. The one Joe was just at, I went to the same one down below campus. It's a good spot to get stuff. Also the turtle pond that's on campus. I know I saw some people there yesterday collecting some things. Indoor location with good lighting. Thermometer and scale are not like immediately necessary, but if you've got them, they're going to be good for readings. pH strips, we are ordering them for you all, but we don't have them yet. So as soon as I hear more about that, I'll let you know. But for now, don't worry about testing pH. And super important when you're setting up, don't forget to blow in that carbon dioxide. The plants won't survive if you don't do that. Just make sure you don't accidentally suck up that gross stream water when you're done. So I would read through all of this, kind of some good background, the biology part of it. Procedure is pretty straightforward. And then once you're done setting it up, you're gonna find, here we go, the measurements part. So this is gonna be due next week with your selfie of the ecosphere. Pretty basic stuff, date and time. Date, time, volume, water, number of plants, so on and so forth. You can either like fill this out on a Word document and upload it, or you can write it down in a notebook. I have all of mine just in here. If you write it in a notebook, just like take a picture of it and submit it. As long as I see that you've done everything that's on this list, you'll get all the points. It's gonna be worth 5 points to have your selfie and your observations submitted next week. There are some at the end, like pH, and there's another one there. pH and weight, these are not mandatory right now because you don't have pH strips and scales. Not everybody has a food scale. If you have one, definitely weigh them out. Joe didn't show himself doing that, but I weighed my plants and my substrate last night. It's good information to have and could help you formulate a better hypothesis with your second ecosphere. You have numbers there, you can adjust those pretty easily. So that's pretty much it for that. I'll have the assignment posted on Canvas by the end of the day, as well as those videos up there already. So yeah, that'll be it. Does anybody have any questions so far on the ecosphere, getting it set up? Let's do anything. All right, let's go. Here. All right, so that's the ecosphere. There's more of those links here if you don't want to get them on Canvas. And I'll have the PowerPoint here uploaded by the end of the day as well. Just talk about all this stuff. Oh, we'll be having check-in sheets at least once per week. So in addition to your initial observations, you're going to have to take more measurements every week to make sure you're taking care of it. iNaturalist, so this was your assignment from last week. You had to get signed up and you had to do 5 observations. Anybody find anything super cool you want to talk about? No, that's okay. You're going to be doing this all semester, so you're going to have to average about 5 observations a week. So it'll be about 60 at the end, and this is a pretty significant portion of your final grade. I'm going to have it set up for now as like 2 points a week to have these 5 observations done, just to make sure everybody's still doing it. It's going to be kind of like a participation thing, but there is a separate participation grade, so I will figure something else out for that. A couple of things I saw. So some people have already submitted INAT observations. I want to go over again what is a wild organism. There were submissions for, I saw a domestic dog, a domestic horse, and a domestic goat, I believe. Those are not wild. General rule of thumb, if you have to feed it or water it, it's not self-sustaining. That is not a wild animal or plant. So please make sure you're finding naturally occurring organisms. Some people posted the carp, I think, from the koi pond, and I'm unclear how I feel about that one. I don't think they're being fed, but they also are not naturally occurring there. But for now, that one's going to be fine. In addition to that, actual identifying stuff. So there were a couple people who just posted pictures but didn't bother identifying what their pictures were. You have to make sure you have identified it. There's like Joe was just saying, there's a suggestion thing, so it'll kind of recognize what it is and give you pretty close suggestions. Mine seemed to find everything that I posted, but you have to have at least tried to do that. You can't just leave that blank. It'd be nice if you could get down to like genus level. Species is excellent. If you get the right species, amazing, but that is not required. Just some sort of identification. Also, if you haven't yet, please follow my INAT account. I'll pull that up again real fast just so you can see it. Just is kind of easier for me to find you all sometimes if I am using that instead. Here. So my name on here is Mitch R. Johnson again. My observations. Yeah, please follow me. That would be great. Any other questions on INAT? About what's going on there? No.
Can you show us how you follow someone there? It's new to me.
Like how you post pictures on here?
No, I have already posted, but you said to follow you on it.
Oh, yeah, I think you just search my name in the search bar and it should pull me up. I've got, I think like 17 students are already following, 18 now. So yeah, just search my name. Mitch R. Johnson or Mitchell Johnson should pull me up.
Got it, thank you.
Yeah, no problem. Oh, one more thing. When you're submitting your assignment for that, so remember you're just submitting a screenshot of your INAT page, so showing me your profile and your 5 observations. Don't submit your actual observation pictures to Canvas. I saw a couple people had done that and had not shown me their actual accounts, so that doesn't really help me. That's just pictures of plants. So don't do that. Any other INAT questions before we move on? All right.
There's one more question in the chat.
Oh, there's, missed that. My chat, that was not. Oh, there's a couple. Okay, where's the creek? If you go down towards the 8, behind the chemistry building, there's a big parking lot. You gotta hop like a little fence to get down there and kind of scale over some rocks. And it's back down there. If you're having a hard time finding it, just use the turtle pond. It's really easy to find. Oh yeah, so let me put some stuff up. What day next week is it due? That's due Monday. So we don't have class next Monday because of the holiday, but your submissions are still going to be due by midnight. So the ecosphere stuff, as well as your next week of INAT observations. And because we're just trying to get like 60 total of the INATs by the end of the semester, it's like an average of 5 a week. So if you're 1 short or like 1 over, somewhere in that range, I'll still give you full like participation points for now. as long as at the end you have close to 60. What else do we have? No, you don't have to do feedback. If we find an index of local species to crossword, that's cool. Probably just Google the index of local species. I don't have any off the top of my head. I can poke around though and see if I can find anything good and send that out on Canvas. Are we allowed to use turtle pond yet? Yes, so you can use the turtle pond for sure. Sweet. Cool. No, that's it. All right, now let's do brief lecture if there are no more questions on our projects. So we're going to talk a little bit about calories and nutrition. And these are relevant to this project as well as the sourdough one, which I'll talk about in a minute, because all of these things are going to be found in both of those systems, especially the macromolecules we're going to talk about. So some basic stuff about calories. I'm sure everybody here has heard of a calorie before. We're going to talk about a slightly different calorie, although they're very similarly related. At a basic level, a calorie is just a unit used to measure energy. Super simple. And the typical calorie that you think of when you hear the word calorie is the US calorie, which is actually equal to 1,000 of the calories that we are talking about, which are the actual units of measurements. So don't mix those up because you're going to get way different values. And a calorie is simply the amount of energy required to raise the temperature of 1 kilogram of water by 1 degree Celsius. Not fair. Basic calorie information, we're going to have little tidbits about how much calories per gram are involved in each of these macromolecules that we're going to discuss. So macromolecules are the things that they're essential to all life, they're in all living things, and they're found in these nutrition facts. I'm going to guess which ones on there are the macromolecules or the biomolecules. Anybody know? All right, let's go over them. Biomolecules. So biomolecules are present in living organisms, and they're essential to biological processes. So one of the main biological processes that come up is cell division. Another big one is metabolism. Super important to all things going on inside living things. We got proteins, carbs, lipids, and nucleic acids. These are macromolecules. There are other forms of biomolecules, but these are the 4 big ones, literally. The other ones are much smaller molecules. These are the ones that are most relevant to our systems. We'll start with proteins. They are roughly 10 to 20% of an organism. That does vary pretty widely, but that's a rough estimate. A protein is a polymer made of amino acid monomers, and a monomer is simply a molecule that can react with other monomers to form polymer chains. And polymer chains are what make proteins. It's the primary structural molecule in animal cells. So animal cells generally would be very fluid without proteins, but they help keep them stiff and rigid. We'll talk more about that with structure. You got 3 main functions in the body. First one is enzymes. Enzymes are molecules that help catalyze chemical reactions. Big chemical reaction there is metabolism, so going back to that. Another one is DNA replication. So enzymes help facilitate these processes in the human or in bodies, in living organisms. Another one is cell signaling. So proteins can be used as both signaling molecules and receptor molecules. And a big, widely known signaling molecule would be the molecule insulin. Insulin basically sends out a signal that promotes the absorption of glucose in the blood. So people who have diabetes are lacking in that signaling molecule and have to take insulin in order to regulate that. So that's an important cell signaling one, and they also function for structure. So like I just mentioned, they give cells themselves structure, in addition to being involved in things like keratin and collagen. So things like cartilage, your hair, your nails, these are all made up of proteins. Cool. Protein function is determined by the sequence of amino acids that make up the protein. So a basic polymer chain is made up of an amino acid sequence, and that straightforward amino acid sequence is your primary structure. Within that sequence, you're going to have repeating sections, basically like building blocks of your polymer chain, and those repeating segments are your secondary structure. Tertiary structure is the actual shape of your overall protein molecule. So that chain will kind of like twist and fold. You can kind of see it in this image over here. So you get an actual shape, and that is your protein molecule. And then your quaternary structure is a few protein molecules together in the shape that that structure forms. Proteins are synthesized in the ribosomes, and heat will denature proteins. So in this example, if you've ever cooked an egg on the stove, the clear part turns white. That is the protein in the egg whites denaturing. And proteins have an energy ratio of 4 calories per gram. Next, we've got carbohydrates. So carbs, I'm sure everybody's heard of carbohydrates before, are just sugars. And they're made up of carbon, hydrogen, and oxygen. And those molecular elements combine to form like a hexagonal structure. And that is a single sugar molecule. So over here, on this image, you can see single sugar molecule, 2 sugar molecules, many sugar molecules linked. So that's those hexagonal structures of carbon, hydrogen, and oxygen either forming together or by themselves. So the single sugar molecule is a monosaccharide, or a simple sugar. Those are going to be like your corn syrups, your white sugar, just basic simple things. It's a great energy source for cells, but it's a very simple molecule. It is not complex in nature. Then you've got disaccharides. Sucrose and lactose are big ones, not as common as monosaccharides or polysaccharides. Polysaccharides are your complex sugars. They are excellent for energy storage in both plants and animals. In plants, that is starch. Probably all have heard of starch. It's big in potatoes. And then in animals, it's glycogen. Less well known, but still important. And these are much better energy storage than monosaccharides simply because of their complex nature. Then it also is used, carbohydrates are used as structure in plant cells. So if you've ever heard of cellulose, that's widely found in plants. Bark on trees is mostly composed of cellulose. Cotton is mostly composed of cellulose. Paper, which makes sense because paper comes from wood. All very high in cellulose. And our digestive systems cannot digest cellulose, but it still helps regulate our digestive system, even though we can't break it down. Other people or other animals can break it down though, and it's broken down by the mitochondria, just not by us. This is the most abundant biomolecule in plants, and in this one, the energy ratio is again 4 calories per gram of carbohydrates. Next, we've got lipids. Many of you have probably heard lipids referred to as fats before. If you've heard of lipids, that's somewhat accurate. They're not synonymous. Every fat is a lipid, but not every lipid is a fat. Lipids are made up of a glycerol molecule and then 3 fatty acid molecules. Common lipids you've probably heard of, especially talking about fats, are saturated fats and unsaturated fats. Classic saturated fats are your cream cheese, butters, coconuts, oils. They're all solid at room temperature. Other unsaturated fats are liquid at room temperature. Those are going to be your oils, olive oils. They're found in avocado and fish as well. Fish oils, super healthy. I know a lot of people take fish oil pills. And these are just determined by the bonds that are happening in the fatty acid chain. So saturated fats have no double bonds in their fatty acid chains. Unsaturated fats have 1 or more double bonds in their fatty acid chains. And they're super important in our body beyond just energy storage. They also are super important for cell membranes. So the next slide, we'll look at lipid bilayers. They also are great for insulating, not just your neurons, they insulate a lot of your organs. And steroids, a type of hormone, they have a type of hormone used in cell signaling. And these have a super high energy ratio. So these are 9 calories per gram. So definitely better for energy storage than carbs or protein. Let's take a look at the lipid bilayer though. So lipid bilayers are great for cell membranes, both on the outside of cells as well as on individual organelles to help separate things inside of a cell. And this works because of a hydrophilic head and a hydrophobic tail. So the way these things get aligned when they're put in water is that the heads want to be towards the water and the tails want to be away from it. And that causes these membranes to form, like this image over here. If you ever put like olive oil in water, you notice that it floats, and that is due to what is going on here. Yeah, so they make great membranes, still allow things to pass through, usually with the help of proteins, but all you need to know about is hydrophilic head, hydrophobic tails, and they form spontaneously in the water. Next, we've got nucleic acids. Nucleic acids are essential to all life. You can't have any life without these. They are your DNA and your RNA, and they are made up of nucleotides. They are responsible for cell division, protein synthesis, and gene expression. So they code for those proteins we were talking about earlier by using these base pairs: adenine, thymine, guanine, and cytosine. They are found in the nucleus, and they are not essential nutrients. So you don't find these on your nutrition facts in food, just because they're not really nutrients. But you do eat them, so you can find them in veggies, spinach, asparagus, beans, peas, your meats, and your eggs, and yeast. So the yeast is what we're going to be using for sourdough. That's why that's relevant there. No, cool. That's your nucleic acids, 1 to 2% of a living organism, so pretty small proportion. Now, let's talk about pH. pH stands for potential hydrogen, and all it is a scale inversely related to the amount of hydrogen plus ions in a solution. So I say inversely because the lower you go on this scale, like down to 3, 2, or 1, the more hydrogen plus ions you have. If you're going up on that scale, up to 14, you have less hydrogen ions. So that makes things acidic if they're low, or basic if they're high in pH. It's essential to biomolecule function, so it's involved in a lot of reactions breaking down these biomolecules. And they're going to be important in your ecosphere. So doing things like blowing carbon dioxide into your water is going to affect the acidity because carbon bonds with water molecules. So you got to be careful. That's one of the things you can hypothesize when you're making those is like if things start dying and you end up getting these pH strips and you find a really high pH, you can maybe blow less in it next time, or maybe change the substrate. Substrate could have a high effect on your pH, find some more basic soil or rocks. And that's why pH will be relevant to ecosphere. Cool, that's it for information for today as far as lecture goes, but let's talk about this sourdough. So in addition to the ecosphere stuff you need for next week, as well as your INAT observations, you have to start this sourdough project. So you have to go on to SciStarter, which I think I showed you last week. If not, the link is definitely on Canvas and again here. And you got to sign up for that. And by next week, you need to have these materials. I'm not going to have an actual assignment for those, but you will have to put this together by later in the week. So I would really advise getting these things on or by next Monday. I will post the procedure for this separately. I'm going to make another module just like I did for the ecosphere for your sourdough stuff. Also go onto this site. Yeah, I showed you this one as well. So this is the actual sourdough starter site. You can find the materials again on here. If you really want to make your own sourdough already, you can just get started. But we are going to do it separately for our project. It's cool stuff. Yeah, so just prep materials. Not a lot you have to do for this. Pretty easy. The big thing for next week is the ecosphere that's actually worth points, as well as INAT. Cool, so homework. I'll get that assignment up by the end of the day, but you remember you need selfie with your ecosphere, and you need initial observations. You know, it doesn't matter how you get those to me, but they need to be submitted as well. And iNaturalist, you need roughly 10 pictures on your INAT by next week. If it's 9, that's fine. If it's 11 or 12, excellent. Remember, we're trying to get to 60 by the end of the semester. And then get together your sourdough starter supplies. So flour, water, container, rubber bands, paper towels, tablespoon. Mostly common household items. If I haven't looked for it in stores for a while, but if flour is still a hot commodity and you're having a hard time finding it, just email me and let me know, and we'll figure something out. Don't forget to register for Size Starter. Please have that done as well by Monday. Does anybody have any questions on what's due, what they're doing this week, or on any of that material that I just covered? Nope. Cool. Do something. Cool. You guys are free to go then. Office hours tomorrow and Wednesday. If you need anything, I'll be there. Or feel free to send me an email if you need any help with anything.
Wait, I had a question in the chat. Sorry.
Oh, yeah. Sorry.
I can just say I wanted to ask if there's any update with the lab notebooks. I think you said that we were going to be like given like special ones or something.
So I posted a module with what we have so far, and I'm sorry it's kind of hodgepodgey, but it has all of the big projects are in there. So the ecosphere is in there, sourdough, I think the COVID one is in there as well. So read through that, but I don't have anything beyond that right now. That's it for now. I will keep you updated once I find out more. If you bought the book that's like on the website, you should return that to wherever you got it from. That's the last information I got on that. Yes,
okay, cool. Thank you.
This PowerPoint will be posted. I'll get it online later today. Anybody else? Nope, that's all the questions in the chat. Cool. If there are no more questions, you guys are free to go.
Thank you.
Yeah, no problem. See you guys next week. Oh, wait, no, you won't. No class Monday.
Oh.
I did have a quick question.
Yeah, what's up?
Hi, it was just about, I had emailed you, but do you prefer email through like email, or do you want us to use like the chat function, like inbox function through Canvas?
So usually the inbox through Canvas goes to my email, but I don't see your
I did it through like regular email. I didn't do it through the inbox through Canvas, but I can if you prefer that.
I would prefer, I think, just regular email. I think that's easier. Yeah.
Okay, because I had sent one and I was just wondering if
I don't see an email from you. Did you get my address right?
Yeah, let me look right now.
Yeah, just double check.
Yeah, so I did the email that says J Newsom. Is that not your email?
That is not me. That's the coordinator. I'm Mitch.
Okay. I'm sorry. That's what I was looking at the name of the email, and I was like, that doesn't even correlate with his name.
Yeah, no, that's the coordinator. That's the guy whose videos we just watched.
Okay, perfect. I'm gonna send you another email under your email.
Okay, so did you find mine? Or you know where it is?
Yeah.
Okay.
Yeah.
Cool. Yeah, that should.
Thank you.
Then. No problem.
Okay. Thank you.
Yep. All right. I'm heading out. The rest of you.
I also had a question.
Oh yeah, what's up?
I just got on the Zoom call like not that long ago, like maybe 5-6 minutes ago. Do I still get points for showing up, or am I just late?
Attendance isn't mandatory, so I'm glad you showed up. But try and be here on time. These aren't going to be very long because they're not in person, so it'll never be longer than an hour.
Okay.
But I'll have this video and the PowerPoint up online later if you look at it.
All right. Awesome. Yeah, I wouldn't mind.
Yep, no problem.
All right. Thank you.
No problem. All right. Bye, everybody.